Recently, the news has talked extensively about how antibody tests are being used in the fight against COVID-19. They're being used to get a better picture of how many people have been exposed to the virus. This information is instrumental in determining an infection rate, which in turn gives a better estimate of the real death rate and understanding where the virus spread was the most severe. It also provides information on approximately what percentage of infected individuals are asymptomatic, information that is key to containing the spread of the virus. A huge benefit of antibody testing is that unlike the traditional nose swab, which can only detect the virus while a patient is still sick, an antibody test can determine if a patient has been previously infected with COVID-19 by detecting the antibodies that the human body forms to combat the coronavirus. However, a major issue with this remains the question as to how long antibodies against COVID-19 stay in a person's system after they have fought off the virus. While antibody tests can be used to help better understand this, if it turns out that antibodies can only remain in the body for a short time, it will make the use of antibody tests in fighting the COVID-19 pandemic far less effective in data collection, as well as greatly threaten the effectiveness of a potential vaccine. One of the major assays used to perform an antibody test is an ELISA, or an Enzyme-Linked Immunosorbent Assay. This is how an ELISA works. To start, the patient's sample is added to the well. If the test is positive, the COVID-19 antibodies will bind to the bottom of the microplate. Between each step, the well is washed to remove any unbound molecules that could trigger a false positive. Then the antigen is added. If the test is positive, the antigen binds to the patient's COVID-19 antibodies and stays bound when the well is washed again. Now the enzyme-linked antibody is added. This will bind to the antigen from the last step. Finally, the substrate for the enzyme is added. In a positive test, it will bind to the active site on the enzyme, starting a chemical reaction that will result in a visible color change. In negative tests, the unbound enzyme-linked antibody will be washed away, and so the substrate will be unable to react, leaving the well clear. The COVID-19 pandemic has been an incredible challenge, and it is one that we will likely contend with for some time. However, as with so many modern problems, it is clear that science holds the answers to overcoming yet another hurdle to mankind. The scientists of the future, our generation, are the ones that will hold the keys to so many incredible innovations.